Hello, my name is Dan Miranda, and I'm a professor at Drexel University. I use Drosophila melanogaster to study disease biology, specifically neurodegenerative diseases and neurodevelopmental disorders. And today, I'm going to talk to you about how you can utilize the fly to model human diseases, strategies and justifications. Before I start, I will say that there are a number of outstanding reviews available in the literature, and I've highlighted a couple of them here on the screen. These discuss some of the common resources available to the Drosophila community, and they also describe some of the common genetic techniques and protocols available which Drosophila researchers often use to study human biology. So, Drosophila is a model organism, and one might ask, why do you utilize model organisms to understand humans? Because models are just like everyone else, right? Well, in reality, we use model systems because the discoveries that are made in model systems are widely used to explore potential causes and treatments for human disease, when experimentation on humans would be unfeasible or unethical. The kinds of experimentation necessary to gain the kind of significant understanding of biological living systems is particularly invasive, and this makes studies on human beings unfeasible. So, what are some model organisms out there? There are a number of them. This is the mouse, Mus musculus, the toad, Xenopus labius, yeast, Saccharomyces cerevisiae, common roundworm, C. elegans, zebrafish Daniorana, and the common fruit fly, Drosophila melanogaster. And there are many, many more. The expectation is that the discoveries that are made in these model systems can provide insight into the workings of other organisms. But why? Well, this strategy is made possible due to the common evolutionary descent of all living organisms. And as a consequence of that common evolutionary pathway, there's a conservation of metabolic and developmental pathways and genetic material across phyla and species. So indeed, fruit flies are like tiny people with wings. A couple of examples of the commonality between structure and function are highlighted here in what's termed structural similarity. So a number of structures in Drosophila organs are functionally similar to structures in mammals and in humans. And so the basic mechanisms required to perform functions within these structurally similar organs are conserved among species. On the right-hand part of the screen, you can see the retina from a human eye. And you see these long folds of membranes and these dark stained kind of puncta. Those are the photosensitive opsins, which are utilized to capture light. On the left, you have a cross-section through a Drosophila eye, and you can see similar laminar structure with folds of membrane here, and you can also see tiny puncta of darkly stained materials. Those are the opsins for the Drosophila retina. And therefore, the basic mechanism of phototransduction utilized in human beings is conserved in Drosophila. So if you can understand how this mechanism of phototransduction works here in this tissue, you have a much better understanding of how it works in this analogous tissue as well. There are a number of structures in Drosophila organs that are functionally similar to structures in mammals and humans. Beyond structural similarity, there's genetic conservation, genetic similarity. And in Drosophila, 65% of human genes that cause disease have a functionally conserved Drosophila homologue. That is, they're doing essentially the same thing in the fly as they're doing in the human. So if you can understand the basic mechanism of function in Drosophila, you can extrapolate that to a similar basic mechanistic function in human beings. In fact, a number of you are probably working on genetic pathways that are mutated in human cancers or in human heart disease, developmental pathways that were initially discovered in the fly. If you can see here on the left-hand part of the screen in panels A and B, that this is a fly wing. This is what a fly wing normally looks like in panel A, and this is what a fly wing looks like when there's a mutation in the gene Notch. And you can notice here at the very distal part of that fly wing a tiny little notch, kind of like a bite out of the end of the wing. That was discovered back in 1914 in Thomas Hunt Morgan's laboratory, and it was named Notch after this notched wing. And now a large number of cancers have mutations in the Notch pathway. Here on the right is an image from a 1980 publication from Christine Nusselheim-Wohlhard and Eric Wietschhaus from Nature, where they're describing mutations in a number of developmental genes which cause aberrant morphology in Drosophila embryos. And the one I'm highlighting here in the middle is called hedgehog. And just like Notch, mutations in the mammalian homologue of hedgehog, which is called sonic hedgehog, is associated with a large number of human cancers. And so if you've been studying either the Notch or the hedgehog pathway, you've been studying a gene that was discovered in Drosophila and whose basic mechanistic functions were initially worked out in the fly. And you can see a survey of human disease gene counterparts in Drosophila here. The point here is not to be able to make out everything on the slide, but to look at the overall number of Drosophila genes that have a human homologue that is associated with disease. So, what are some of the common strategies that Drosophila biologists utilize to study human disease? Well, many researchers have heard of the power of Drosophila genetics, and this means that flies can be utilized in two distinct genetic ways. They can be used for what's called reverse genetic schemes, and that means you start with a human gene, you find the homologue in a fly, you then make a mutation in that homologue, and then you try to understand the phenotype based on that genotype. Once a human disease gene is identified, you can identify fly homologues, create mutations, analyze phenotypes, but it also allows you to bring in a number of extraordinarily powerful genetic techniques that are adaptable to use in Drosophila. These include tissue-specific targeted knockdown. So if you want to only knock down your gene of interest in the nervous system, we can do that. Or just in the retina, we can do that. Or just in the liver. We can also utilize what's called clonal analysis, and that will allow you to make very specific mutations that are expressed only in a population of cells that you want to express that mutation in. CRISPR-Cas9, the fly is amenable to that genetic technique, Galfor UAS system, the Markham techniques, and we can also express human transgenes. So we can actually take a human gene, we can put it into a fly, and we can express that human gene or human mutations within very specific Drosophila tissues. And I'll discuss some of these techniques in more detail. So the Galfor UAS system is an extraordinarily common genetic technique which is utilized by a number of Drosophila researchers to study human disease. And this allows you to temporarily knock down a gene of interest in a specific tissue of interest. So you can control both time and the tissue where you want to either express or knock down this gene. And this is performed by taking a fly and expressing with a yeast protein which is called GAL4 that I'm highlighting here. That GAL4 protein is under the control of a tissue-specific enhancer which will allow expression of GAL4 at a particular time in development and within a particular tissue. GAL4 binds to what's called an upstream activator sequence, or a UAS. This is also a sequence which is not normally found in Drosophila, but rather is put in through a transgenic animal. And then that GAL4, once it binds to this UAS, will turn on the gene downstream of GAL4 binding. So that can be a human gene, or that could be, for example, a RNA interference construct which creates a double-stranded RNA which could be utilized to specifically knock down a Drosophila gene in a tissue and temporal specific fashion. So it's a bipartite system which allows you to study this gene, or control rather, gene expression. And what you're seeing on the screen now is an example of a genetic scheme that will turn on green fluorescent protein specifically in the embryo of Drosophila in this kind of striped pattern that I'm illustrating on the screen. GFP is downstream of the upstream activating sequence, and GAL4 is downstream of this responder element that's going to specifically express GAL4 in those stripes. Once GAL4 is expressed, it binds onto UAS GFP, and then GFP is only turned on where GAL4 is expressed. To utilize this to study human disease biology in Drosophila, researchers tend to express human proteins with this genetic technique. For example, in Alzheimer's disease, researchers, including my own laboratory, express the human form of the amyloid precursor protein, or APP, and or they will express the human mutated forms of APP. And they'll express this specifically in the brain or specifically in the retina or other areas of the nervous system. When we express human APP, specifically in the adult Drosophila nervous system, we've been able to show that we're able to generate flies that produce A-beta 42 puncta, which are consistent with aggregates found in human Alzheimer's disease brains. And that's illustrated here on the lower left-hand panel. In A, this is a wild-type Drosophila brain. And in B, this is a Drosophila brain, which is expressing human amyloid precursor protein. And you can see these tiny puncta here. Those are small aggregates of the human APP protein. We were able to stain this brain with antibody that will specifically detect amyloid precursor protein. That's in red here. It's called 6E10. And then also with a fluorescent dye, which is a derivative of Congo red, which allows us to detect the presence of amyloid structures, and that's in green, illustrated here. And you can see that there's significant overlap between both antibody staining and amyloid staining, which is consistent with what we see in human disease biology. We can feed these animals drugs which specifically block the formation of the amyloid beta plaques, and we can see that animals fed on that drug, illustrated here, have a significantly decreased number of puncta as opposed to control animals that are not fed on that drug. And this is useful because we can then utilize this to screen through a large number of therapeutic drugs in an in vivo system. We also utilized the system when we showed that this model has memory deficits. It forms amyloid structures in the brain, as I've just shown you, and is responsive to therapeutic intervention. Another genetic technique heavily utilized in the study of human disease is called the Markham technique. And this also utilizes the GAL4 protein, but it also uses a second protein, which is called GAL80, which is also from yeast. And GAL80 essentially shuts down GAL4 whenever they are co-expressed. When they're both in the same tissue, GAL80 wins. And it allows for the creation of what are called genetic clones with highly specific genotypes. This has been extraordinarily useful in cancer studies because it will allow you to create homozygous loss-of-function mutations in small population of cells within an organism, thus mimicking the etiology of cancer. This is performed through forced genetic recombination, whereby you have a single chromosome, illustrated here, that has a mutation of interest, say a mutation in a tumor suppressor gene or an oncogene, and then you are also expressing a GAL80 within that same cell. Now you're expressing a GAL4 in that cell, and then an upstream activating sequence followed by some marker. In this case, that marker is GFP. GFP will allow you to visually identify which cells carry or harbor that mutation. Because GAL80 is being expressed within this cell, the GAL4 is shut down. That is, it's not activated. Through normal mitotic division, we can force recombination within these two chromosomes, forcing crossover at these sites right here, which will create sister cells that now have a copy of this chromosome, which carries that mutation, and because of genetic recombination, a copy of this chromosome right here, which also harbors that mutation. If you'll notice, the recombination also creates two chromosomes, this one right here, which carries GAL80, and again, because of recombination, this chromosome right here, which also carries GAL80. Now you'll notice that in this cell, this daughter cell, GAL80 is completely removed, but GAL4 is still present, and that allows GAL4 to turn on. In this daughter cell, you now have two copies of GAL80, and so that GAL4 is shut down. Because GAL4 is turned on, it binds to this upstream activating sequence and turns on green fluorescent protein. You can now take that animal, put it underneath a fluorescent microscope, and identify that cell by the presence of the green fluorescent protein that's glowing. If it has GFP, it also has this homozygous mutation of interest. And then you can now study the effects of that mutation in a clonal fashion. So this has been, as I've mentioned, utilized extraordinarily successfully in cancer biology. Here's an example that I'm showing you where researchers have activated the oncogene RAS in Drosophila, and they've coupled this oncogenic activation of RAS with mutations in the Hippo pathway, which is a cell adhesion pathway that is highly mutated in a number of human cancers. You'll see here in panel A what a wild-type Markham clonal expansion looks like. You're seeing a larval stage here, so what you don't see is actually the whole rest of the worm or the tiny little larva, but you can see the little green cells here that are highlighted with GFP. If you now simply activate RAS via this oncogenic mutation, which is found in humans, you see there's no significant overproliferation of the GFP clonal tissue. If you only mutate the hippo pathway, specific components of the hippo pathway as illustrated here, you also don't see a significant expansion of this GFP tissue. However, if you both activate RAS and knock down hippo, or cause mutations in components of the hippo pathway, you can see a significant overproliferation of this clonal tissue, as indicated here. And you can also begin to see, if you'll notice where my cursor is, small areas of green fluorescent protein, or GFP, that are present outside of the initial clone. And what those are cancerous tissue, or cancerous cells that have metastasized away from the initial oncogenic event. And thereby you're mimicking human metastasis through this genetic technique. So what these researchers were able to suggest is that loss of the gene Scribble, which is a component of the hippo pathway, in combination with oncogenic activation through RAS, results in invasive and malignant tumor growth. And this is also the case in human cancers as well, and it was first worked out here in Drosophila. So those are some examples of reverse genetics. Drosophila can also undergo or use forward genetics, which is starting with a phenotype and then attempting to screen for genes based on this phenotype. You can also screen for drugs, and you can correlate those drugs back to genes. And this strategy has been heavily utilized to identify new targets and therapeutics in disease exploration. This, similar to reverse genetics, allows you to utilize the power of Drosophila, including Markov analysis, Gal4 UAS systems, CRISPR-Cas9. But this also allows you to bring in Drosophila behavioral analysis, because you can utilize behavioral screens as the basis of the starting phenotype. And then you can look for genes or drugs that alter this behavior. Because Drosophila are invertebrates, they don't require IACUC protocols for any of these screens, and this opens up the options for very quick proof of concept tests and analysis. An example of this was performed in Mel Feeney's lab, where they utilized the Gal4 UAS system to perform an RNA interference screen. And what they did here was they expressed mutant form of tau protein, and the mutant forms of tau are associated with familial frontotemporal dementia, and they expressed this mutated form of tau specifically in the developing eye of the fly. You can see here, this is a normal-looking adult Drosophila eye, and this eye right here is a fly eye that is expressing this mutated form of tau. They then systematically evaluated 67 different candidate genes based on Alzheimer's disease-associated genomic loci from published human genome-wide association studies. And they found a number of cell adhesion pathway mutants that were important for Alzheimer's disease susceptibility. And they did this by looking for modification of this specific eye phenotype here. And you can see in panels C through J, co-expression of RNA interference transgenes to knock down these 67 candidate genes in the same tissue as expressing this mutated tau causes a enhancement of this phenotype. And this nicely illustrates the power of Drosophila for functional genetic screens following up on human GWAS studies, moving from humans to Drosophila. And one laboratory that utilized this particularly well comes from Ross Kagan's laboratory. Ross Kagan was able to identify Kelpresa which is now an FDA-approved chemotherapy for RET-based thyroid cancers. RET is a human oncogene, and what Ross Kagan did was express a specific human mutation of RET in the Drosophila eye, as you can see here. So here in panel A is a normal wild-type looking eye, whereas in panel B is an eye expressing a mutated form of RET. And then he screened these animals for drugs that were able to modulate RET expression. And they were able to utilize the Gal4-UAS system. They were able to use a specific property of the Gal4-UAS system to modulate the amount of RET expression within this developing eye. And that property is temperature sensitivity of the Gal4. So at lower temperatures, Gal4 is less active, and at higher temperatures, Gal4 is more active. And so by utilizing Gal4 expression, they were able to tweak the amount of expression of this RET mutant. They identified Calpresa, and in 2010, that was FDA approved as the first therapeutic intervention for RET-based thyroid cancers. It was a big day in Drosophila biology. And I want to highlight the Ross Kagan Laboratory just one more time, because what they're doing now is they're taking this same technique, but they're kind of turning it up to 11. They're really doubling down on the usefulness of this technique to identify patient-specific drugs or drugs that are specific to an individual patient. And how they do this is utilizing the Gal4-UAS system, as previously shown. And they do a biopsy of a patient's tumor. They then sequence this tumor and analyze it. And they identify the handful of mutations which cause this cancerous phenotype. And usually this is 12 or more mutations which ultimately cause this cancerous phenotype. They then create what they call a personalized fly avatar, where they create transgenic animals or transgenic elements that contain the exact same mutation from the human tumor, and then they put that mutation under UAS control in the fly. And then they turn on all of those mutants in the fly, and then they screen this highly complex phenotype, which is based on this human cancer, for drugs. After they find drugs which suppress this mutant phenotype in Drosophila, they move to targeted therapy in humans. And they do this by taking a piece of the biopsy from the patient and putting that into a mouse, giving that mouse cancer, and then taking the drugs that they identified by screening the fly and giving it to the mouse to see what happens to the tumor in the mouse. And if it shows good efficacy in the mouse, they give it to humans. Drosophila can also be utilized for models of axon injury and nerve damage. This comes from Nancy Bonini's lab at the University of Pennsylvania. Here she expressed a membrane-bound green fluorescent protein to an axon that spans the outside part of the Drosophila wing, as you can see here. This is actually expressed within one of the wing veins, and this is a long axon that travels all the way down to the distal end. And what the Bonini lab did was simply take scissors and cut the fly wing. Flies don't need these to live. And by cutting the fly wing, you're also cutting that axon. And then they were able to screen this phenotype looking for drugs that could modify this effect. And they identified the nicotinamide mononucleotide adenyltransferase, NMAT, that they suggested is required to maintain mitochondria after this particular injury. Drosophila can be used to study heart disease. And a unique aspect of using flies to study heart disease is that you can defibrillate a fly heart for minutes, and you can measure the response of this defibrillation in a living animal over time. Here is an example of a heart, and this is the same example of a heart that is in defibrillation. If you defibrillate a mammal for a couple of minutes, that mammal will die. This is also useful for modeling channelopathies and cardiomyopathies in Drosophila. And there are many, many, many more ways of utilizing Drosophila. The neuromuscular junction is one such model. The NMJ in flies is glutamatergic, not cholinergic like it is in mammals. It's also amenable to electrophysiology, the same kind of drug and behavioral screening that I just showed you. An example of using the NMJ to identify disease relevant human biology comes from the Shaker potassium channel. This is affected in many human diseases, including cardiac arrhythmias, deafness, and epilepsy. And this was first discovered in Drosophila. A giant fiber system, you can use this to analyze neuronal function over time, unlike the NMJ. And this is helpful for understanding epilepsy, seizures, by using high-frequency stimulation in the eye and then detecting seizure-like electrical activity in the muscles. Optical coherence tomography, or OCT, this is a non-invasive kind of echocardiogram in Drosophila that allows you to perform live imaging of the heart, and this can also be used to measure heart rhythmicity. Behavioral assays, including learning and memory assays, motor assays, climbing and contractions. These are useful for studying cognitive dysfunction and are also amenable to behavioral screening and drug screening. You can also use Drosophila to study diabetes and diseases of insulin resistance, Huntington disease, ALS, Parkinson's, developmental disorders, including CHARGE syndrome and Pitt-Hopkins syndrome, two disorders that I studied in my laboratory. Fragile X, sleep, cancer, repeat expansion disease, inflammation, infection, kidney disease, metabolic disorders, alcohol disorder, and many, many more. So, if you have any questions, please contact me. That's my email address right there. And I would be happy to tell you more about how we can utilize Drosophila to better understand human biology.